So, in the previous video, we talked about the different things that antibiotics might do to target bacteria, and we saw this slide. And basically, what we're saying here is we're targeting things that have to do with the central dogma of biology, again, which is the DNA to mRNA to protein, and that incorporates these two things. And then we also have other crucial functions like cell wall synthesis, membrane structure and function, and folic acid synthesis. But what we're going to focus on right now is the central dogma, and in particular we're going to focus on translation and how certain antibiotics will inhibit that. So this is a nice image that shows us all kinds of different targets for different antibiotics in the cell. So you could come back and look at this in a little bit, but you'll see that we do see cell wall inhibitors, membrane inhibitors, DNA and RNA replication inhibitors, and then we also see the inhibitors for translation. So, if you recall, translation is a process that is mediated by the ribosome. The ribosome is a molecule that all cells have. So, any living organism has a ribosome, or has many ribosomes. However, as we talked about in the previous video, the ribosomes for bacteria are distinct from those in mammals. And this is a summary chart, so this is a lot, I know, but if you're a numbers person, then you might want to check this out. And this is just quantifying how different the bacterial ribosome is from the mammalian ribosome. And so you could see here that they have different masses. The mass of the mammalian ribosome is higher. Interestingly, the bacterial ribosome has more RNA in it than the mammalian ribosome does. And the separate subunits, so the large and the small subunits, are also different masses compared to when you compare bacterial and mammalian, and we call them by different names. So the bacterial ribosome, you might hear the large subunit referred to as the 50S and the small subunit referred to as 30S, whereas with mammals we have the 60S and the 40S. Now the S is something you might not have heard of before, but it's just a measurement referring to the mass and the density of some molecule. And so it's just used to describe the different characteristics of those subunits. Again, you don't need to know all this stuff. I just wanted to show you that the molecules are indeed different, and the antibiotics are going to bind to one and not the other because of those differences. And then there's a little summary of that down here as well. So what exactly is it that antibiotics are going to do? So we have another cartoon of a ribosome here, and this is again a bacterial ribosome. So we have both subunits. They're not distinguished, differentiated here, but you could imagine this is where they are. And what happens is that the antibiotic will only be able to bind to the target molecule, that is, the molecule of the bacteria. And in the case of an antibiotic binding to the ribosome, it can do a couple of different things. So it might bind in slightly different places. So again, remember the ribosome has 3 different binding sites, or 3 different sites where we have tRNAs coming in or existing. And antibiotics can bind to those sites, or they could bind to other parts of the ribosome. And different antibiotics do different things. So for example, chloromethylphenidate It binds to the 50S portion, so the large subunit, and inhibits formation of the peptide bond. What does that mean? What is the peptide bond? Well, that's the bond in between the amino acids. So essentially, if this antibiotic is bound to this ribosome, you cannot have the formation of a protein. You're just going to get an amino acid, or rather a tRNA coming in, but then it can't, the ribosome can't add that amino acid to any others. It can't link them together. So you can't make any proteins. Of course, the cell can't survive if it can't make any proteins. Erythromycin, which you might have heard of before as a treatment for yourself or others, binds to the 50S, again, the large subunit, and prevents this whole subunit from moving along the mRNA. And then we have tetracyclines, which you might also have heard of, and tetracyclines interfere with the attachment of the tRNA to the mRNA. So in other words, it interferes with the codons matching up with the anticodons. And so essentially it just gets in the way so that those two things can't match up. And then finally we have streptomycin over here, and streptomycin binds to the smaller subunit, which is the 30S, and it is going to interfere with the mRNA being read. So that's just an overview of a couple of different types of antibiotics, and these are all antibiotics that are only targeting translation. Of course, there are antibiotics that target other things, but we're not going to go over all of those because there are a lot of different ones.